Okay, thank you all. Appreciate the time. I know it's been a long day, and it's always hard to be toward the end of the list because everybody's tired of listening to things. But anyway, so my name is Cowan. I'm at the Medical University of South Carolina. This is my first Champ One meeting. I want to thank you all for the invitation to come and speak. I met Jeff about 5 years ago. He found me on the web because I study neurodevelopmental disorders and I'm a researcher. And it's been really gratifying to sort of see how this has all evolved. I got him in touch with Stefano, and it's really cool to kind of see how this research project has grown. In my rush to put the slides together, forgot to put an acknowledgement slide at the end, so I'm going to mention people, I hope, as I go through, but I wanted to point out my graduate student, Alain Greige, who is somewhere. There he is, Alain over here. And I just wanted to point him out. He's really a brilliant graduate student, and it's really his brainchild that really got us thinking about this potential therapeutic approach. So I want to be sure and give credit where credit is due. Okay, so what I'm going to try to do is I can go quickly through some of this stuff because you've already gotten a background, but as I was putting this together, I wasn't sure what you would have heard about yet. And let's see if this will work. I'm pushing down. The blue light's coming on. Hold on, let me see if I can get it started. Maybe it's just sleeping. Not going. Right now? Yeah, okay, perfect. Okay, so a couple of quick disclosures. This idea I'm going to tell you about today is something that we have a patent application pending for a related neurodevelopmental disorder that I'm going to set out for you today just to give you a flavor of where we're going with this. We're just getting started on the Champ One, and I'll even describe a little bit about the POG Zeta possibility because we hadn't really thought about this too much, but I know you're all going to ask me at the end of this talk. So we have the method for identifying and deploying these TSBs, as you'll hear, for MEF2C haploinsufficiency syndrome as well as for CHAMP1. As you know and have heard, as you were thinking about brain development, you start with cells in the brain, like you heard from the last talk from Daly, and those cells function, they wire up into your brain, and they form a functional network that allows us to experience our world and adapt to our experiences. And within those neurons, obviously we have in our nucleus, we have DNA, and that DNA encodes many genes. In our average neuron, we think we're probably somewhere on the order of about 13,000 genes or so that are expressed. And the expression of those is often controlled by proteins called transcription factors. And so these little things that are sitting on the DNA, and as we've heard from several talks today, whether or not that DNA is open or closed helps determine whether that gene's going to get turned on. And one of the genes that we've been studying for many years, now more than 20 years my group has been studying, is a transcription factor family called MEF2C. And MEF2C proteins, they form dimers. That means they bind together in a group of two. They bind to your genomic DNA in specific sites, and they turn on specific target genes. And they don't do this by themselves, but they do this together with other factors that I'm just going to show as a little green blob here and call co-activators. And so these transcription factors turn on a series of genes, and as you've heard a lot today, they give rise to messenger RNAs. And this is, I always like to think of this as, this is the blueprint, or this is the instructions here. This is, the photocopier now has made instructions and sent it out to the work site. And then out, as you get out, the protein product, which is the final product, typically is what actually makes the cell function and do all the things, a lot of the things that we think of. But in addition to this sort of canonical view of transcription factor turning on mRNA, which makes a protein and the protein does something, there are also a huge proportion of our genome that codes for RNA that does not give rise to protein. And those are called non-coding RNAs. And those non-coding RNAs are thought to be potentially produced from as much as 60% of our genome, whereas the protein represents a much, much smaller proportion. And just as a little aside, through human evolution compared to lower species, the non-coding RNAs have really exploded. And so it's thought that those non-coding RNAs that don't go on to make a protein, they come back and they interact with these messenger RNAs and they affect the expression level of the proteins that are going to be produced. And we think that that gives rise in part to some of the variety of differences between humans and lower organisms. Okay, so the disorder we've been studying for many years, and the way I'm going to outline this talk is I'm going to walk you through this idea based on the studies we've been doing with this MEF2C haploinsufficiency syndrome, and at the end, we're just getting started on the CHAMP1 side of things. I'll signal where we're going and what we're trying to do for that. So this idea we had a number of years ago, about 2010 or so, a number of patients were reported in the literature with autism symptoms, as well as a number of really commonly occurring symptoms, including seizures, intellectual disability, hypotonia, a lot of the things you see with CHAMP1 syndrome. In fact, when you read the symptom profile, it is shockingly similar. Now, that's not totally surprising, because actually a lot of neurodevelopmental disorders, particularly monogenic ones, do have a number of these features, but it is really, really similar, interestingly, which may just be coincidentally. So most of these patients who was originally reported were deletions of the MEF2C gene. So that's why it was determined to be a haploinsufficiency syndrome. However, now that we have more than 400 patients worldwide that we're aware of, and more every day, we're realizing now that we have missense mutations, we have frame shift mutations, we have micro and macro deletions, et cetera. All of them very, very, very similar symptom profile, which leads us, at least in the MEF2C side of the world, to think that a lot of these are probably loss of function. And in fact, we've tested this directly through a number of patient mutations. Okay, so almost all but one that I know of the 400 so far, are de novo. De novo meaning that neither mom nor dad had this mutation, but probably arose through a mutation that happened in the egg or the sperm, and the child inherits this mutation. Now, this is a haploinsufficiency, so MEF2C is not on a sex chromosome, so we have one wild type copy and we have one mutant copy that is transmitted, that the offspring carries. And these kids are pretty severely affected, and partly we think because MEF2C is an incredibly critical gene for brain and immune system development, actually, turns out as well. Okay, where was I going with this? I'm rambling a little bit. Okay, so MEF2C is found on chromosome 5, and it's at the 5q14.3 portion of the chromosome, and again, there are two copies of it. In a neurotypical human, you would have each of them producing messenger RNA and protein, and which would be very similar to what you see with the CHAMP1 and POG-Z, I assume. Where I've forgotten which one of them is X-linked, but in the case of deletions or mutations, then one of those copies is not being properly generated, and so often, particularly with deletion, you're just losing half of the protein, the messenger RNA protein that would be produced. And so as we were working on this for many years, and we started thinking, well, one way we'd probably try to help understand this is try to generate an animal model that would give us some insight. And so, part of the goal here, the thing I was going to tell you, part of the goal, obviously, is that we took a very simple-minded approach, which is that perhaps one way we can try to help families with MEF2C haploinsufficiency is simply by increasing the expression of MEF2C. Being agnostic to what MEF2C does and in which cells. We know the fundamental problem. There's not enough MEF2C from one copy in order for the body to do what it needs to do to have neurotypical development and function. So we started, just like we heard earlier, with many years ago, generating an animal model. We generated a mouse where we've deleted one copy throughout development. And interestingly, these mice present with very robust behavioral phenotypes. And we see this across a lot of features. They show almost an absence of social preference. They generate many, many fewer vocalizations. They generate these ultrasonic frequency vocalizations that humans can't hear, but we can record. They show evidence of some repetitive motor behavior. They're extremely hyperactive. They show changes in the elevated plus, elevated zero maze, like we heard earlier. They show abnormalities in sleep, in terms of sleep structure and sleep function. And we see lots and lots of differences in the way that the cortex wires and is functioning. So we have a pretty good animal model for this, and we've really gone a little farther, and we know that it's expressed in a lot of different cell types, expressed in neurons, excitatory neurons, it's expressed in inhibitory neurons, it's expressed in blood vessel cells, it's expressed in the immune system as well. And so the brain functions as a complex interaction between all these components, and we could genetically delete it, so to speak, or remove one copy from all these different cell populations. And we see a subset of the sort of symptoms in the mice, if you will, if we take it out of different components. So what that tells us is that it is functioning in multiple cell types, that the reduced function is contributing to the behavioral phenotypes that we see, and the synaptic phenotypes in part, through a combination of different cell components. So then, if we're trying to generate a therapeutic approach, how do we know which cells to target? And how do we get it to the right cells at the right time, and so forth and so on? So we really started brainstorming about this, and again, we came up with this very simple-minded view that we know the problem with MEF2C haploinsufficiency. There's not enough MEF2C from the one surviving allele to overcome the loss of the other mutant allele. So, as we were thinking about therapeutic approaches, where my lab has increasingly started moving over time, we decided to try to tackle a potential therapeutic that would try to increase the expression of MEF2C. And we started with a viral approach, much like you heard earlier today. That has some challenges, getting cell type, getting the right expression level, et cetera. And so as we were thinking about all the various things that we wanted to take into account, we started thinking about the fact that, you know, MEF2C is not expressed in every cell at the same level. And it's not expressed in every cell at the same time. There's dynamics through development. Also, the dose probably matters. We've learned this through Rett syndrome, that loss of function of MECP2 causes Rett syndrome, but duplications of MECP2 cause a syndrome as well. So too much or too little of many of these genes can be problematic. So how do you hit that just sort of right dose, if you will, in order for the brain to function and develop properly? The other one that we don't talk about a lot is something called alternative splicing. So MEF2C is a really long gene with lots of exons, and the way that, you know, I showed you a nice little messenger RNA, but the reality is every cell makes a slightly different version, and even the same cell may produce multiple different varieties of that same gene. And so how do we decide which one of those splice isoforms is important and which one can we upregulate? So what Alain and I started thinking about is different ways that we could try to tackle this. I just wanted to point out that CHAMP1, interesting, unlike MEF2C, which is really quite different in different brain regions, CHAMP1 seems to be a little more uniform across the brain if you look at different brain regions, what you're looking at here on this profile. But if you look within cells, and again, this is data that we pulled up from the web. We haven't personally generated this data, so I can't verify all this is totally accurate, but it looks like neurons express CHAMP1 at higher levels than microglia, which are the brain's immune cells, and then at the lower end, astrocytes and oligodendrocytes. So we need to hit a lot of different cell types, potentially, and maybe the amounts in those cells need to be modulated appropriately. So this made us think about how a way that we could intervene and potentially upregulate and modify MEF2C. And so one way we tried to do this was trying to interfere with the naturally occurring process I was referring to a moment ago, which is these non-coding RNAs that are naturally produced in all of our cells that feed back. They don't make a protein, but they feed back and affect the messenger RNA stability and expression. So these microRNAs are small pieces that are processed in the cell, again, from non-coding regions, and they're typically about 21 to 23 nucleotides long. They do a lot of different things, but one of the things I really want to point out is that they bind to the messenger RNA to regulate the translation, which is the movement from the messenger RNA to the protein. So it binds the messenger RNA and it keeps it from making a protein at the same rate. And by doing that, it limits the amount of protein that you produce. So if we can interfere with that, we might be able to have a chance to modify the expression of a gene of interest. Okay, the other thing is it's really involved in tissue-specific cell expression. So this is a way that we could potentially go into any cell type and not really know or care how each cell is supposed to be regulating MEF2C, but let the cell tell us what to do, but simply by interfering potentially with regulatory mechanisms along these lines. Okay, sorry, I'm belaboring this point. So here, in the simplest cartoon, this is a messenger RNA. Now it's just already been spliced, and so we'll just pretend there's only one spliced isoform. This dark part is going to be the protein coding portion. Oops, I think Alain even has a cartoon here for me. So, you go from this mRNA to this protein. And this part here in the dark, this is the part that's going to code for the protein. Then there's all this other stuff that sits on the end, which is called the 3' untranslated region. So most genes, most transcripts will have this bit, and it doesn't have anything to do with the protein. But it's there to help the cell determine how much to make. And it does so, again, by a feedback mechanism. So these are those microRNAs that I was talking about, these small non-coding RNAs, and they will bind to regions often of untranslated region. And when they bind, they limit the expression of the protein through cellular machinery. And so in this particular cell, in this particular case, this microRNA is bound to this mRNA, and the silencers are going to slow the translation process, and you're going to result in less protein being made. And this is an important regulatory process, again, that is found in every cell and for almost every gene. So how could we potentially interfere or interact with this mechanism as a way to develop a therapeutic? Well, one of the ideas we considered is something that folks in the field were considering called an antagomer. So we could introduce a small piece of RNA that binds to this microRNA and acts as a decoy. And so basically binds this thing, doesn't allow it to bind to MEF2C. So now MEF2C can make more protein. Now that can work, and there are certainly folks out there who are pursuing this approach. But one of the negatives is this same microRNA can bind hundreds of other genes potentially and regulate lots of other the expression of lots of other proteins. So there's the potential for off-target effects, if you will, or side effects as an indirect consequence of regulating other things other than MEF2C. So the other way that we can tackle this problem is something we're calling a target site blocker. Now this target site blocker is a little different, but really taps into the same mechanism. And again, a lot of credit to Alain for really coming up with these ideas and pursuing this. But the target site blocker, again, now we have a small piece of RNA that is chemically modified, so it's extremely stable. It can last in the system for 3 to 6 months, and the endogenous machinery of your cells is unable to effectively destroy it and clear it, which if you had a normal messenger RNA, if you put a normal RNA in there, it's not going to survive for that long. So it's chemically modified, but it doesn't elicit an immune response, which is important. But by doing this, now you have the target site blocker binding. This microRNA can't bind and regulate the expression of MEF2C. And as a consequence, hopefully, you get more MEF2C protein. So does this work? So the way we went about doing this is, of course, we looked at the 3' untranslated region of MEF2C. We dug deep and looked to see all the different places that microRNAs are known to bind or are predicted to bind based on those that have been already, that we know exist, these non-coding RNAs, and we identified a number of them, and we sort of prioritized them based on whether they had stronger neuronal expression or immune system or muscle cell expression, et cetera, and we picked a number of them to test. And I'm going to show you the results from one of the first one we tried, which you can see here. This is called a Western blot. This is the amount of MEF2C that you find in the cells. When you treat with the target site blocker, you can see we get an increase in expression of MEF2C. Now it's not huge, right? It's 30–40% increase. But remember, we've lost about half. We really probably want about double so that we'd have the same amount as a wild type cell. And we don't know where the threshold is. Maybe 30, 40, 50% increase is sufficient for the cell. We don't know where that threshold of having quite enough. Okay, so this one worked. We tried another one. It works also. This one works a little better, about 50% increase in MEF2C levels. We've now tried about 9 of them, and all 9 of them work to varying degrees. And we're now pursuing different, because there's a lot of sort of optimization of this to try to figure out what's sort of the best strategy going forward. Okay, so it works. Those are cells in culture. that we see that are human cells, and we've seen them in multiple types of cells. We think that this is likely to work. Karim Al-Hassani, who's a postdoc in my lab, has been working on this project together with Alain. And so we took the first target site blocker and we injected this into the intracerebral ventricular area. So, your ventricles in your brain. And we did this in a newborn mouse. We injected in, and then we asked a couple of questions. First, we said, how long do the target site blockers last? And if he looks, a little hard to see here, sorry about that. If he looks 50 days later, so about 2 months later, we can clearly see evidence that the RNA target site blocker is still present in the brain of this animal. So that's great, right? I mean, it seems to be distributed broadly, and it seems to be enduring. The next thing he did is we said, well, does it actually work in vivo? Does it work in the animal? Can we get upregulation of F2C? And so what he's been doing, and this is kind of where we are in the project right now, he's injected it. We've looked 10 days later. He takes the brain out. We do a Western blot. We look to see levels of F2C. And in this particular mouse, a series of mice, actually we've got quite a few mice now, we see about an average of 65% increase in F2C levels. So again, we don't know what that magic threshold is for how much we need in order to get therapeutic benefit, but in principle, twofold would be perfect. Maybe, but probably less than twofold is going to be sufficient. We'll have to see. Okay, so what about CHAMP1? So I approached Jeff about 6 months ago or so, and I said, this looks like it's really working well from F2C. CHAMP1 haploinsufficiency could be a great candidate for considering this. And I don't want to get into the weeds about or an argument here about what gain of function, loss of function, et cetera. But with the sort of general idea that if we can increase CHAMP1, this could be potentially beneficial. We've identified 9 microRNA binding sites that are sitting in the 3' UTR of CHAMP1 messenger RNA, and we've now synthesized 5 of the target site blockers with this modification, and we are testing this currently in our human cell lines. We're working with Stefano Bertot, so we'll be testing this both in sort of wild-type cells, cells as well as we even have cells from these two, the nonsense patient mutation and the frameshift patient mutation. And as you've heard from Stefano or from Daly earlier, we can detect CHAMP1 expression really nicely. So we can see if it has an effect. Here's the CHAMP1 knockout. You can see that the protein is gone. So this looks very promising. And hopefully, and this is the data courtesy of Stefano's lab group, who are sitting over here in this table right here. So the other thing I wanted to point out really quickly is that in addition to the potential, and again, just potential, I don't want to give false hope here, this may or may not work, but the potential to upregulate CHAMP1 through this target site blocker effect, this could be combined with other RNA-based therapeutics, such as the one you're going to hear about some from Dr. Buxbaum later, where we could be looking at antisense oligonucleotides that would be targeting patient-specific mutations. Those two things can happen in parallel. You can be increasing the wild-type non-mutant copy and mutant copy and simultaneously targeting the mutant copy for degradation with the thought that simply removing the mutant copy may not be enough, because we do know that there are deletion mutants in the CHAMP1 patient population who have symptoms. So clearly, you're going to need more CHAMP1. Okay. And then I knew I was going to get this question, so we threw this slide in about 2 hours ago. Could we do this for POGZ? Of course. This really could work for almost any gene. We looked briefly. There's about 21 microRNA binding sites predicted in the 3' UTR of POGZ, and these could certainly be tested to see if we can get some effect there. So I'm going to stop there. I want to thank again Alain, Karim Al-Hassani in my lab, and the Bertot lab for the ongoing collaboration, and I'll take any questions.
Thank you very much. Oh.
Great talk. I have a question. Would you ever consider a combination of microRNA targeting site to confer specificity and really increase it to twofold instead of just 1.5?
Yeah, that's a fantastic question. I'll try to answer it as succinctly as possible. Yes, we've considered it. We've even tried it. Actually, we've combined two. Interestingly, we don't get a whole lot more than not much more of an increase by adding two instead of one. Probably because we may be saturating the cellular effect of the microRNA repression. It's just a guess. We've been talking to the FDA about getting FDA approval for trying this in human patients, and what they told us is that if we can do it in one, that's ideal, because otherwise every single one of these that you try has to be tested as a separate drug. So we would have to do the full safety profile, et cetera, for each of them separately in order for them to be used combined. And that's going to be true also for a combination of a TSB with an ASO as well. Each of them has to be shown to be safe together. So our dream is that we find one that works best. And I think that's what the advice we're getting from our brain trust.
Right, Chris, over here. So lipid nanoparticles. I'm just wondering if, you know, we've learned from the Moderna vaccine or these other vaccines that, you know, delivering some of these things might be done best in a lipid nanoparticle. Have you thought about that?
I have, actually. So there's a number of really things we're interested. So I didn't get into this, but the way that this would be delivered, if we're trying to target brain cells, particularly from F2C, is intrathecally. And what that means is an injection into your spinal cord, into the spinal canal, to get it through your cerebrospinal fluid to bathe the brain. That is a routine thing that can happen at any doctor office, so that's not a big problem, but it's going to have to be redone every 3 to 6 months. Ideally, we would want something that could be delivered through the vein, right? An intravenous infusion. And so there are a couple of different approaches we're trying to take. One is to potentially take these target site blockers and conjugate them with proteins like the transferrin receptor that would get past the blood-brain barrier and allow us even potentially to do some specific targeting. But also we're working with some collaborators at the University of Florida who have created a lipid nanoparticle onion, if you will, where you just layer the RNAs within more and more layers of lipid. And what that would do is it gets into the cell is obviously release more and more over time. And so you're exactly right. And the cool thing about that approach is that you can coat the lipid nanoparticles in principle, even to have receptor binding specificity, right? So you could maybe even get some cell type targeting. Sorry, this is into the weeds, but this is where I live.
Any other question?
And I'll be around tonight for dinner if anybody has follow-up.
So, thank you very much.